Welcome to the You Be The Chemist session today here. I'm Larry Cernick. I've got also co-presenters Kerr Meow and Marcia Gillette, who will be doing some fun chemistry activities with you. I'm going to start off. I was a scientist for the first part of my career, and then I became a project manager, but I really enjoy science. It's been very passionate about it. And today we're going to do some fun chemistry activities. Now I want you in, if you can, and Kermit, I'm going to open the chat window myself too. I'm going to share this camera here. And I've got a cup here of a clear, colorless liquid. And I'm going to waft it, and it's got no odor. And it is a liquid. What do you think it is? Type in the chat what you think that might be.
Someone say water. Abby says water.
You think it might be water? Okay.
That's from Abby, yeah.
Okay. Yes, and that's it is some water, but it's also got a certain percentage of hydrogen peroxide. Here's the bottle that came from. So it's this is like a 3% solution. So it's 3% of hydrogen peroxide in water, and we're going to do something fun with hydrogen peroxide and show you a nice chemical reaction that you can do with that. Now to do this, and there's you can download from the website there the list of supplies that you need. To do this, you want to do it on a table, probably your kitchen table would be a good place to do it, and you need one of these trays. And you want to have something that's pretty deep because it can flow over. Then you're going to need a 16-ounce soda bottle, and it says just, the instructions just say put it on the tray, but you see it's not very stable. So I purchased, and you could make one of these at home. This is available on Amazon, maybe some other places. It's basically a bottle holder or cup holder. So you put your bottle in there and it makes it nice and stable so it doesn't fall over. Okay, so what the next step is you can watch it as we're putting stuff into that. I want to get so you can see what's going on in the bottle. You need a funnel, and what we had here in this cup here is the 3% solution of hydrogen peroxide and water, and I'm just going to pour that into the funnel and into the bottle. You want to have that. So it's just, again, hydrogen peroxide in the bottle. Then we're going to add a couple of 2 teaspoons or 2 to 3 teaspoons of dish detergent. I'm going to use Dawn, but other ones will work too as well. Just pour that in there. This is blue color, if that's okay. You can use white if you've got white, or clear, I should say. Clear works. It's not going to really have much effect on it there. So all we got in the bottle there, we've got now hydrogen peroxide solution plus soap, and then I'm going to add some food color. Today we're going to do green. And what you want to do is on your bottle, you want to get the liquid food color. You want to drip it down the side of the bottle. You want to drip like strips of it down the side of the bottle and put a couple of those so it goes all the way down the side. Don't just dump it into the bottle. It looks nicer if you make the strips down the side of the bottle. Okay, so we got food coloring there and hydrogen peroxide solution with dish detergent on top of it there. Okay, now comes the magic. So what you need to get is some yeast. Your mom probably has some of this active dry yeast that you can buy. She uses it for baking there. You can buy the little jar of that there, and that'll last you quite a long time. You want to make sure it's good and fresh yeast because it loses its activity over time. So what I did is I made this in advance. You can see it's actually puffed up quite a bit. It was just, so I put some 2 teaspoons of the yeast in there, and then I added some water. The water needs to be of temperature about 110 to 115. If you go too high, you'll kill the yeast. And if it's not warm enough, it doesn't work as well. So now what we're going to do now is add this to the bottle, and then we're going to watch what happens. gotta swirl this a little bit here. It didn't mix very good. There we go. Now watch what's coming up in the bottle there now. Now if this activity is called elephant toothpaste, and you can see it's going to become more apparent as it comes forward. There are some other enzymes, catalysts, I should say, that you can use to make this reaction go very, very quickly, but they are not very safe to use. Yeast is very good. You can do it at home, and so you don't have to worry about that. It doesn't give you as nice a display there. It's coming up. It's going to be flowing over the top in a second. So if you look at the side of the bottle, I'll show you there, you can see it's growing towards the top. There's a lot of bubbles here. So here's your soap and water and hydrogen peroxide, and then it's rising to the top there. There's something coming out of the hydrogen peroxide and water, and it's coming up towards the top, and it's eventually going to flow over the top. So this is an example of a chemical reaction here. It's actually a decomposition reaction. I'm going to show you the formula for it. And here we go. And this is why they kind of call it elephant toothpaste, because it flows over the top like it's toothpaste coming out of a tube there. And it's a large, long stream of it. So that's why they call it elephant toothpaste. Okay. It's definitely not as dramatic when you use the yeast, but it's still pretty nice. Okay, I'm going to put that aside. I'm going to just show you the chemical formula for what happened here. So this is again called elephant toothpaste. You can download the activity from the where you connect it to this experience there. So it's a chemical reaction, actually a decomposition. So we have H2O2, which is hydrogen peroxide, which is a liquid, and it's dissolved. There's some water in there too as well. It breaks down to give 2 H2O2 gives 2 waters plus 2 oxygens, and the oxygen is a gas. And it's very important that what the soap does for you is it captures the bubbles, the oxygen bubbles, and then comes out of the bottle there too. What the yeast does is that it has an enzyme called catalase, which basically acts as a catalyst to speed up the reaction. Normally, H2O2 does break down to water and oxygen very slowly over time. But with the catalase added, it goes much, much more quickly. So, okay, so that's a fun activity you can try at home there. I'm going to just share one other thing, and then we're going to pass it over to the next presenter. If you're in grades 5 to 8, you probably can't see it too well there, but let me see if I can zoom in on that. Should be able to zoom in on that. There we go. A little bit better. Okay, there's one of the programs that is put on by this group that made that activity there is called the You Be The Chemist Challenge, and it's a competition for grades 5 to 8, and there's study materials online. You can, between now and December 31st, you can register, and just go to www.chemid.org and programs and challenge, and you can find out all this information there. So you can register now. The study materials that you would use for this here, it's a team-based competition, and then there's also a video competition that's going to take place between November 2020 and April 2021. You can find out more information on that. So this is the registration. Here's the actual competitions for the teams. It's going to occur in February and March, and then there will be a national competition in May to June. It's a lot of fun. It's all virtual this year, but in the past it's been live event, but this year it's going to be virtual, and I think it should be a lot of fun. Okay, that's it for me.
Okay, I think that's my turn. So can you hear me, Larry?
Yes, I'm
Okay.
going to have to, yeah, okay.
Can I start, or you need to do something?
Yeah, no, go ahead. I'm going to have to leave because I got to make sure the other sessions are working good.
All right, so good morning, everyone. My name is Perm Yao. As you can see on my camera, my name is right there. Professor Gillette and Dr. Cernick are both friends of mine for many years. We all use science as our career. We also like science because it's so much fun doing science, not only for problem solving, but also for activities like today. That's why we are here with you today, trying to show you some fun activities that you can do at home safely, but at the same time, learn a little bit about science and have some fun about it. So today's my topic is science is art. Science is very regimental, very fixed. We do this for problem solving, but sometimes we'll find some ways to make it very fun and beautiful. So that's what I'm trying to show you today. How can science be art? So the material you need is very simple. Some of those actually, Dr. Cernick used it already. All you need is some milk from home in your fridge. Whole milk is the best, but if you have other milk, potentially they will work too. And Dawn Ultra dishwashing liquid, and Larry used it too, so you already have that. Food coloring, and Larry used that as well, so that means you can also use that. Don't let it go to waste. And the fourth thing is some cotton swab. Sometimes we call them Q-tips, something like this in front of the camera. If you have those, that's perfect. And couple plates that are waterproof. That means the liquid won't drip everywhere. That's all. That's not what you want, and not your mom and dad would like to happen. So find some waterproof paper or plastic plate. Okay, so let's try it. See what we can do with these few things. So as I said, I have a plate of milk here in front of us. So it's just white, right? Nothing happening. So let's see what we can do about it. So we talk about food coloring. I buy this from the store. You can find it in any supermarket or dollar store. Very easy to find, very inexpensive. They come with 4 different colors. The more color you have, the more fun you will be. So what I'm going to do right now is just that with the food coloring, I just put a few drops in the middle. Make sure your milk is stable, so don't shake it. Let it stand for a few seconds. That's all you need, 10 seconds. And then put a few drops and then do something around. I have 4 colors, so I would use all 4 colors. So put a few drops in the middle and then some on the side. You can do whatever you want. The pattern is up to you to decide. There's no special way to do it. The color is just there for you to see. And a few drops of red in the middle and then some on the side. And one last one, I have blue as well. So see what happened with the blue. Now you see that when I put the dye into the milk, nothing happened. They just stay there. But now I use my Q-tip. I dip it into the Dawn detergent. So this is the one that I got from the supermarket. Everyone has that at home. Somehow Dawn seems to work well. But if you have others, I will encourage you to try it. Now what I'm doing is that I pour some into a glass and then dip my Q-tip into the detergent. Don't let it drip, just dab a little bit on the side so that it's not dripping. That's all you need, a little bit on top of the Q-tip. Now pay attention, see what happened when we put it into the center of the milk. Whenever you move the Q-tip to a different place, the color starts changing direction and moving away from the Q-tip. The longer you leave it there, the more pattern will show up in your milk. Isn't it cool? So you can go back and use the other end of the Q-tip and dip it into the detergent one more time and try again. So what's happening here? Why is it happening that way? So we have to ask ourselves, what are we seeing here? So let me take that away and then show you my my paper one more time. So why don't you go to the chat and share with us what you see, what you observe? Can you use 1 or 2 words to describe what you see on the milk? Anyone? Good comment, Abby. The soap reacts to the milk to push it away from the Q-tip. That's very good observation. That's exactly what we would like to describe when we see something happen. We want to describe it in word first so that we can capture it on paper or on a computer. Okay, now we have to help ourselves understand what is happening, right? So what we use is called the scientific method. When you go online, go to a textbook, go to a chemistry book, when you think about color and milk and movement of color in milk, you will find these 2 terms, surface tension and surfactants. And when we see something happening, we want to go and research a little bit. What are these all about? Get some background information. And then you will find that these 2 terms are relevant to this particular experiment. And then you come to think of it. Is that the milk has some surface tension on it? Is it the detergent have some surfactant characteristic or soup characteristic that will break the surface tension? Is it what this is about? So we form a hypothesis because all scientists want to guess what exactly is happening. So that's what we call hypothesis. We form a hypothesis to help us answer the question, to solve the problem, or to explain the situation. So in this case, my hypothesis is that milk potentially have high surface tension from some of the molecules. We call them fat molecule. I'll explain that later a little bit. With this surface tension, the color doesn't move at all when you drop the color into the milk. But when you put the soap into the center, the soap break the surface tension and start moving the color away. That's my hypothesis. Now I want to test if this hypothesis is true. Is milk has high surface tension that can be broken down by soap to allow the color to flow. And then we design an experiment. When we design an experiment, not just to do what I just did, because that will help me to see the observation, but I want to make sure that this is true. That's why we call it the control experiment. Now I show you what to do, what I meant by control experiment. Let me move that and bring 2 new plates in under the camera. Now you can see that in front of me, I have 2 plates. One on my right-hand side is just water, so it's clear, right? The other one is milk, just like the last dish. Now I will do the same thing using the food coloring. So few drop here and few drop there. Make sure I have all the color because this is the only way to make it pretty. Now, once again, once you drop the color into the milk or into the water, they don't move because theoretically, based on our hypothesis, there's surface tension. The surface tension just keeps the color together without doing anything. So now they are all localized in the center, both in the water and in the milk. First, I use a Q-tip that has only water. So I have water here. So this is Q-tip. I dip it into some water, just plain water. Now, see what happens if I just dip it in water and dip it in the center. Uh-oh, nothing happened in the milk. If once again, I put the Q-tip in water and put it on this side, on the water side. And once again, there's nothing happening. So that is what we call the control. That means water doesn't work, Q-tip doesn't work. It has to be more than just Q-tip and water. So what do we do? Use a new Q-tip, put it into detergent, the Dawn, dab it on the side so that it's not too much. Now let's try see what happened in the milk. Ooh. Now I use a different Q-tip, a clean one. I put it into the detergent, and now I put it into the water side. See what happened. You see a little bit, but the color doesn't move that much, even with the detergent in the water side. Let me try one more time. More detergent. Let's repeat it. Sometimes do science. We want to make sure that we repeat it and the result is consistent. That's why we do duplicate or triplicate to make sure the data is what we want to find, not just a fluke, right? So now we did 4 experiments in total. So let me move that away and then show you what I want to, how we collect this data, right? I don't want to spill everything on my table. Okay, so we finished the control experiment. This is the data we want to collect. Just a very simple 2x2 table here. Let me grab my pen. So we have used 2 different cotton swabs in water or in soap. And then we have 2 plates of liquid, water and milk. When we have water and then we use water cotton swab, we do not see any result, negative. Same with milk, but use water cotton swab, you saw negative result. And use cotton swab with soap in the water with color, we kind of see a little bit. It moves quickly and then it's gone. But if we use soap inside milk with the coloring, we see a lot of color movement. So that's how we capture the data, right? Using the table. With this table, we can see all the results. We did 4 experiments very, very quickly. And now that's how we capture the result. Now, with the result, we can explain the data. So color flows freely in the milk when we broken it with the soap or the surfactant. Remember the term surface tension and surfactant? Soap serves as the surfactant that breaks the surface tension of the milk. Once the surface tension is broken, the color allows to move freely in